Welcome to today's lecture. Today we will continue on the protein comparison part. I ended last Thursday with a slide from the paper from Rationale et al., which says there are different structural alignment methods, and somehow they all have strengths and weaknesses. And if we could find the best of all, that would clearly be the highest of these curves. Let's get back now to protein space and databases. For many years, the way I imagined the space of proteins is like this, where essentially a protein structure, often also referred as the fold, would be one island. The coloring here implied that for some of these islands, the green ones, we know the structure, and for some, the white ones, we don't. And there are some islands that are a little bit green and a little bit white, indicating that there are regions in this protein for which we don't know the structure, and there are regions for which we do. Maybe the entire fold or the main fold we know, but not everything. And in some sense, one of the goals of structural genomics was to do a structure for every single one of these white islands. How did pieces evolve? How did proteins evolve? There's a variety of assumptions about that. One assumption, one view comes from Andrei Lupas, who is at the Max Planck Institute in Tübingen. Andrei Lupas argues the world life started with RNA molecules. RNA molecules cannot do their own catalysis. So they need something to help them to do the enzymatic activity. What could that be? Now, being an RNA molecule wandering around in the soup of those days, you have the idea, well, amino acids, right? They come next to you. And in fact, you put amino acids together, peptides, and peptides can do the catalysis for the RNA. In that view then, peptides, proteins, or protein fragments, pre-proteins if you want, are the slaves for the RNA life. That then would imply that the types of amino acids you use are the ones that help RNA to survive, RNA to bind. That would imply that the folds, the types of shapes we see that are done by these 20 amino acids are selected according to their ability to bind nucleotides. And if true, you would expect that some of these original activities, some of these original shapes, you may still see in today's structures. In some of these cases, they may, just like in this case of the particular coil, they may just be two helices that stick like that and fit into a groove. They may be helix-turn-helix motifs, or they may be repeats, or they may be in this propeller here more than just two strands, just one sheet. They may just be this propeller motif. And that could be then something that is smaller, shorter than domains, that evolves independently in some sense, and that actually was what started it all. How would you find out whether that is true? Well, you would try to see, staring at alignments, what is common between them, and is it true that those original motifs go back in time, and can I actually go into the lab and make them? Can I build structures from these original motifs? And they have been doing that for 20 years. And the story they built, when you hear Andrei talk, is very impressive, very convincing. He really has put his life's effort going from the young guy to the bearded guy in the course of it into making this point, and I'm, as an observer, convinced that he's onto something. That certainly seems to be one path. Maybe there are others, but I believe this is one convincing story. Nick Grishin from the University of Dallas, HHMI, Howard Hughes Medical Institute, comes with a somehow similar story. By just staring at the evolution of structures and sequences, he argues that maybe the way we move is not in domains, not in folds. So this image that I gave to you before, that there are islands and each island is a fold. Maybe it's not true. Maybe there are fragments, and maybe part of it is in fact this sort of beta strand motif here, the Greek key motif, and then you fold into helices, you fold into other things, but this is the original motif, and maybe that is the point. If so, then I would look at many structures and find these motifs. So this is not necessarily driven by an assumption about evolution, but it is driven by the assumption that there are some aggregates, some local units that could be helix turn helix, so 2 secondary structure segments. There could be more than that, but they are clearly short of the domains, and they could be the units in which evolution moves, or at least one of them. Again, Nick may not claim that this is the entire truth, but again, when we look at his work, we see that the motifs he finds, and here just a few examples, he can build very convincing stories. If that is the case, and we clearly believe more truth to this than to our old idea of folds, then there are no islands. Then there are no folds. Then if essentially, at least, the idea of an island, if you think about it, is you can't go from one to the other without a ship. So imagine that you smear this out, and that in fact the way you can go between islands, you can just walk, like when there is no water. When there is a big ebb, you can just walk from. And what I mean here is you can essentially move from one structure to another by just a few changes. Maybe you can't reach the entire universe this way, but you certainly can do more than we thought. You certainly no longer believe in this idea of folds, and this idea of islands, and this idea of units that are written in stone. Okay? That's a very important statement to make for me, because for the remainder of the next, at least for the next 40 minutes or something, I'm going to show you something else. Namely, the view that there are folds. There are units, and that we can classify proteins by these units. Why do I do that? Well, ultimately, it helps to guess similarity in function. It helps to learn about evolution. One thing is clear, we need to classify. We need to find things that are, there are two approaches. It's like Plato and Aristotle. So Aristotle, in some sense, the Aristotelian view is that you collect a lot of items, you find similarity between these items, you put a label on these similarities, put them in a box, put a sticker on the box and say this is X. And that's understanding. By just sorting everything that you see in front of you into these boxes, by putting a label on it, you acquire understanding, just by seeing what is similar to each other. The Platonian view is there are the ideas, so that there's a basic understanding, there are basic principles. So the way we understand the world, that's the sort of physicist versus biology, if you want. The way we understand the world is by understanding first principles. Okay? This is biology. Proteins are biology. We need to begin by collecting, because we don't know how a protein folds. We cannot take a sequence, put it into a computer, and get a 3D structure. As long as we cannot do that, we don't understand protein structure prediction, and we don't understand how folds form. We may imagine that we do, but as long as we cannot predict, we fail. Okay? As long as we fail, we need to classify. We need to revert to the biologist's view of it, and that means put stickers on it. That means we need units to put into our boxes. Back to folds. Christina Rango, I'll get back to her later, has this view that, again, there is a common theme, and upon that common theme you model what she calls embellishment, additions. Proteins are just common themes and a lot of additions to it. Now, one issue here, is it really true that there are a limited number of shapes? So, how would we answer that? Or let me pose the question slightly differently. So, there's the universe, or there's the space of protein sequences. And there's the space of protein structures. And let's assume that the width of that circle that I draw depends on the diversity of groups that I have in one or the other. Sequences, structures, do they have the same width, the same diameter? Because you believe there are more, so...
Because you have different values and...
Aye. That's a very good answer. I see what you say. In fact, so you're touching the point that is exactly my goal here. Let's put it differently. When we look at different genes in human, or at different proteins between us and mouse, very often we find that there's a protein in human and mouse, they're very, very similar in sequence, yet they are not identical. You can easily see that there is maybe 90% sequence identity between two proteins and two different organisms. They have the same function, they have the same structure, and. So by this logic, these two would classify into the same family of sequence, but they have the same structure. The structure is more similar than their sequence. So these structures would be more similar than 10%. Let's put it like that. So they're both recognizable as families here. One family here, one family here. But now if I said, okay, 10% difference is already something. Let's get to the structure. In many of these cases, for 90% sequence identity, in fact, I will not observe the residue, that 10% of the residues are in 3D in a different position. Beyond, okay, now we get into what you said. Ultimately what you said allele, but you could have also even said one particular structure is about motion. So not everything in the structure is fixed. Here's a loop that moves more. So you shine light onto this thing twice at two different time points, and you will see slightly a different structure. Do I call that a different structure? No, it's essentially one structure, okay? As long as I call that one structure, I can now look at, say, the protein in mouse and the protein in human, and ask for the 90% of the residues that are in fact equal in sequence, do they have more variety than what is anyway the protein moves around by itself, right? The human 100% has a variance. Is the variance between human and mouse higher than that? And the answer to that question is no, typically. So that's one way of saying, okay, the structure of the human and mouse is more similar than their sequence. That is saying that the space of structures is more condensed. Or is saying that the variety of sequences, there are many, many sequences that fall to the same structure.
So there would be an argument for a limited number of shapes then?
That would be an argument. Well, now we get into biology again. It is certainly limited, because the number of proteins is countable. It's not infinite. And certainly by this argument, the number of structures would be smaller than the number of sequences. It could still be fairly large. Let's look at what we have today, and that brings us back again to the issue that we have to accept that there are folds for a moment. And then we can put that concept down. But what we already see in this argument, it all depends on whether this loop moving or not, I count the same as the same structure. So ultimately it has to do with what do I call a different sequence. So the variety between two people, is that a different protein? No. By the way we define it, we have a sequence variance, but we call it the same protein. The same protein is not always 100%. Okay? And you immediately see that we get into a stretch, what we call a protein if the protein does not always have the same sequence. Okay, we can say for that person it comes from the same region in the genome. But you see immediately that the story gets more complicated. And a similar story here in terms of structure. But I believe we can agree that there will be one way of defining similarity that is acceptable. Now, on the most extreme end of the classification, back to the Leavitt classification here, we simply see the universe as all alpha, all beta, alpha, beta. So those are very disjunct units. Okay, there's a tiny alpha helix in here, but essentially those are very clearly distinguished objects. How do we recognize the similarity? And in fact, here I'm going to present 3 different approaches how to recognize similarity. The first one is called SCOP. And SCOP comes from Alexei Moltsin, Cambridge University. About 90 publications, 1 over 3,000 quotes, an age index over 30. This in no way reflects the importance of Alexei Moltsin. Alexei Moltsin, how does he classify structures? Well, originally, he just came and looked at it. That was at a time when the PDB had 20,000 structures. He knew every single one of those. Okay? He could look at it and would immediately tell you, in a blink of a second, I've seen that before. When you gave him a structure that was not in the PDB, that was similar to something in the PDB or not, he would immediately still come and say, well, I've seen that before, or maybe not. And then he would go into the computer and play a little bit around with it. But ultimately, he would say, yes, I was right or I was wrong, but he immediately gave you an idea why this is the same, why it's similar, and he looked at these. Have you looked at structures? So he looked at these things, at long structures, big enzymes, Tim barrels, and said this Tim barrel is related to that one. He knew nothing about the sequence, just looked at the 3D object in no time. So he has an incredible visual memory. And he mapped this visual memory on the ability to understand an intuitive understanding of protein structures. And that is in fact the way he classifies structure originally. So he, in some sense, then rationalized what he did. He's a scientist. So you have an intuition for how it's right, and then you put a logic upon this finding that you have to argue why you were right at the first place. Because it's very difficult to say, this is right because I say so. This is not going down very well with reviewers. So he puts it into a logic. The logic is a hierarchy. And the hierarchy begins here. Alpha, beta. That is in fact what SCOP calls the class. Now, we go increase the similarities. Those are the first, is not as simple as I showed you before. On the first level of classes, we have slightly different things. So one here, originally you see multi-domain proteins that are classified together, and that brings us back to the issue of how many proteins have multi-domains. That was done at the point at which PDB, the proteins of known structure, essentially has only one domain. Membrane and cell surface proteins, small proteins because they fall out, coil are these. Low resolution protein structures, so essentially that means we can't say. Peptides, short things that we don't want to classify. Design proteins, I don't know. That's the logic behind it. But you see that there is something from structure, driven by structure, to something that makes sense in the context of structures. Then, on a level of SCOP class here, you have the Rossmann fold here, and the Tim barrel, the 2 different types of alpha-beta structures that you even now you can immediately see look different, recognizably different, and they should be classified by both are alpha-beta, but they're clearly different in terms of the way the helices are grouped around the central strand, where here it forms a hole, a pore if you want. Here it forms more of an elongated structure that is sandwiched. On the next level, so all alpha, then this is essentially the level of globin-like. And this level, Alexei then called the fold, because that was started, SCOP was started 20 years ago, when we all still believed folds exist. Now, the fold definition here is we have the same major secondary structure, so essentially you have helices, you have the helices that are alpha-beta in the same succession, the same arrangement, with the same topological connections. So that now means you could have the 2 alpha helices like this, and another one 2 where you go up here and down here, or you could just cut the loops and connect them the other way around. That's the topology. So fold defines topology. So when you cut the connections, that's no longer the topology, because actually, if you imagine that the protein folds, you cannot reach the same. Protein cannot just cut the loops and to swap around, right? So fold is driven by this idea, this is the way a particular protein folds, okay? The peripheral, what Christine calls the embellishment, the peripheral elements here, they may differ. Now, Alexei then put the rationale into what peripheral elements mean. So the moment peripheral elements become too big, the argument is that the fold no longer is the fold. And that brings us to, this is before the idea of Grishin or Lupas, in which we have fragments that are short. This is still based on the assumption that the unit of classification is a fold, is an entire thing that folds on its own. Single domain is something that is... the evolutionary relation within a fold is unclear, meaning that between two proteins that have a similar fold, you may not be able to say that they diverged from a common ancestor. Okay? Example for a fold, timbarrels. Here are 3 different timbarrels, and appreciably, in particular in this orientation, they are very similar. I don't remember the level of sequence identity between those, but it's fairly low. They are fairly different. And we get back to that in about a week from now. So, now, again, I just wanted to make this point here. We don't believe in fold anymore, but it's a way to start. On the next level here, so this is on the level of structural relation only. This is on the level of I say something about that protein. Superfamily. Anybody heard of a superfamily? The notion of a superfamily? Well, in our regular life it doesn't happen. I heard it first from Christina Rango and Janet Thornton. I'll get back to that in a minute. And Scott copied it. Both of those are in Cambridge. So maybe I should turn these slides around. But SCOP really is driven by structure. That explains this way around. So superfamily is the same thing as a fold. Again, the axis here, they're more similar. 2 cases in a superfamily have more similar structures than 2 cases in a fold. They're recognizably similar, but the differences here are smaller than there. But the difference now is that we assume, Alexei Murtsin assumes, that he can recognize an evolutionary connection. In that sense, family. Superfamily is not easily recognizable. That's what it says, is a big family. So again, probable common evolutionary origin, low similarity, but same fold, and most likely have the same function. Again, here we have the timbarrels. In this particular case, not clear. I should have put some more numbers and compared them to each other. So on this level here of a superfamily, they are more similar. In this particular case, they are also related in sequence. And then we get to the next level here, the family. The family really is a set of proteins that are recognized by their sequence similarity to have a similar structure. The superfamily are typically diverged so much that you cannot recognize their similarity unless you see the structure. But from sequence alignments, you typically could not see that. They would be timbarrels. While in the family here, it's a very clear-cut case. Everybody knows, everybody sees, so in the olden days people put the more than 30% sequence identity, so more than 30% of the residues are identical. But not always. For instance, in globins, some of the cases have much less. Let's go. Ultimately in the head of Alexei Murtsin, with a lot of computers in the past, using on top. Here's Cath. Cath, here's the website, comes from Christina Rango, UCL, of 120 publications, H-index over 40. Christina Rango, I mentioned her briefly with Willie Taylor. She implemented one of the first structural alignment methods, SUP. That's one thing. The next thing she used that for is Cath, and she did that with Janet Thornton, who is still, until July, the director of the European Bioinformatics Institute in Hingston, Cambridgeshire in England. Did an undergraduate study in physics. is in several occasions mentioned as one of the top 100 scientists in England. Over 400 publications, one with over 11,000 quotes, H-index over 88, and you immediately see we get into the giants here, and Janet is a true giant in the field. This is the slide from 97 years now, almost 20 years old, that in some sense for me started it all. So Carth started to classify, and one of the first Carth publications here in Structures in 97 just made a slide with all the structures that then existed. In 97, this is half of that slide. But immediately you sort of recognize the diversity of structures. And this is ultimately, let me go back again to this argument about fold. When you assume that there are folds and that you can use those folds to base your classifications on, then what you are rewarded with is beauty of divergence. And then you see these amazing shapes. Well, most of you have seen these. I always like this horseshoe here, for instance. Anyway, Carth, as the name may reveal, uses 4 levels of classification: class, architecture, topology, and homology. And now you can already guess the way it works. They both come from England, from Cambridge. Carth, the first is the regular class, just like in SCOP. Architecture. Now we get into classification according to overall shape, but ignoring connectivity. Topology, that is in fact fold groups. So if you remember SCOP, the fold is somewhere in between these two. So fold has connectivity in it, but this is sort of more detailed. So this has already more conservation. This is more generalized than a fold. This is just a similarity of a 3D object. Completely removing the connections mean that what you base your comparison on is only the 3-dimensional object, not the way it evolves. That's a valid way of doing it. That has built in it this idea that folds don't really necessarily have to be, because you really begin from structures. You're back to fold somehow here, and you're back to evolution somewhere here. And again, Carth invented this terminology of superfamilies. 4 years ago, or 3 years ago, those were the numbers. About 1,200 shapes, folds, about 2,000 superfamilies, or 2,500, 11,000 sequence families, and 24,000 domains. And you already, in some sense, see exactly what I said before. The sequence universe is larger than the structure universe. Depending on how you call the structure universe, whether you make the cut here, the 12,000 shapes, or you say superfamilies 2,500, 2,500 shapes. But this all has 100,000 sequences under it, at least for the PDB. And I'm not sure whether I get into that, but the largest superfamilies, the very largest 300 superfamilies, I believe about 30% of all the sequences that we know fall into the 300 largest of these superfamilies. So superfamilies 2,500, but 300 of those are really huge. Huge. Tim Barrel is an example for that. So the way Carth then has to evolve, step number 1, you begin to parse the domain. And one particular way of Carth is that it sort of combines expert and machines. Alexei and Mozin was already taken away, so they couldn't take Alexei's head anymore, so they needed computers. And find domains is done by, in this particular case here, 3 different parsing methods. So they all take a 3-dimensional coordinate structure and parse the structure into domains. When those 3 methods agree, the argument is clear. Clear-cut case, automatic deposition. When they disagree, then this is flagged for somebody to look at. Okay? This is what I mean, a little bit of expertise combined with a machine. Unfortunately, completely consistents are only 1/5, so they had to sort of slightly alter that. But they found a way to slightly alter that so that they don't have to look at too many. So that's the domain parsing. From the domain now, once we have the domain here, we get to the level of the superfamily. And the superfamily level, we have conservation in sequence, and we have conservation in structure. So we begin with all the ones that have a similarity in the 3D shape, and then we map the sequence back on top of it. Yeah?
Can we expect function similarity in superfamilies?
Yes. So yes and no. So in typically the assumption is that a superfamily would be enzymatic. So everything in there would tend to be an enzyme. The specificity of the enzymatic activity may differ within, or does differ within a superfamily. So the assumption typically is that you go down to the level of smaller than a family, and then you get the specific enzymatic activity. On the level of the superfamily, you in fact, it may be there may be hydrolases. Even there, some superfamilies go beyond the hydrolases, but I'm not aware that there are superfamilies. I would have to ask Christine whether there are superfamilies that completely are not mix enzymes and non-enzymes in the enzymatic activity domain. I do not believe that's the case. So in that sense, they would be similar in function. So for the Tim-Barrow fold, again, is another fold that I believe we have seen only in structures. But there are several structures, several 3D motifs that you see in different functional connections. So the level of fold, that statement would not be true. That statement tends to be true in the superfamily. And in fact, so this word gene3D here is a link to a database that annotates for all the sequences their three-dimensional structure with a rationale of ultimately functional classification. So let's put it like this. Christina Rango spent the first 10 years of her life doing the 3D classification. Then they got these huge superfamilies, and now they sort of take them apart to identify what's the difference in their function. So they begin with a superfamily and now cut it apart again, but within that superfamily. That's the way they do it. Okay, so Cath versus Scop at 80% residue domain overlap. So what I mean here is that I say a domain in Cath and a domain in Scop are the same if 80% of the residues overlap. Okay? I'm not doing an alignment, I just ask, so both of them call it something a domain. Do 80% of the residues that they call a domain agree? If so, then I say they agree, and that in fact I have for 70% in the PDB that essentially is dominated by single domains. Okay? Now, that is a sort of more detailed map here. I don't want to get into. I believe we should go back to that level here. Does that mean they agree?
Is the question, does that mean they completely the same overlaps?
They're not, right?
No.
But is that so? Maybe I should have done this differently. I should have, before I showed this slide, I should have just asked you what I expect. And would you have expected more or less than that? So there are two conditions, mind you: 80% and 70%. So if I require that all the residues are the same as 0, or very close to 0, I actually don't know what the number is, but it's pretty small. So you have to allow a deviation of 20%, and then you still. Let's ask differently. If I asked you between human and mouse, so we both have roughly 20,000 proteins, okay? And I asked at what level of sequence identity, say at 80% overlap or 80% sequence identity, how many proteins would I find similar between human and mouse? Less or more than 70? I believe is the question. Do you have any feel for that? Anybody has any feel for the genome similarity between man and mice? That's one of those things you can find easily in Google. By the TV. So rumor has it that mice and men are 90-something, 95, 90-something% identical. That may or may not be true. But certainly on the level of the protein, that is not true. And in fact, I do not know the answer to my own question. But what I do know is that if I allowed myself, so if I, instead of 80% here, went to 66, then this number would be 66. So, meaning that this is way, way higher than the similarity between human and mouse proteins, which we typically believe are very similar. They're similar enough so that for all the human biology we study mice. And yes, there are limits to that procedure, but there are lots of benefits. Mostly that goes right. Most of the drugs that essentially exist today work because this worked. Some of the problems and the side effects come because it doesn't quite work. But what we have, we got only because that does work. Okay? So in that sense, this is relatively similar. But in the other sense, and you begin with that statement, it's not completely similar, so there's diversity. And that is diversity on a level before the domain parsing problem becomes an issue. On the level of a superfamily, and this is 50 structures here, you don't quite see that now. Yes, in this particular case, this is in fact dependent, there's a typo, aspartate aminotransferase, like fold, and in fact these are all aminotransferases. 50 different structures, and again you see that there is now helices here, the strands behind. You see that there tends to be a little bit more conservation in the strands, so the strands you can see much clearer somehow than the helices, but even the helices are remarkably similar here between something that has again I would have to look at the diversity of sequences here, but that is one enzymatic activity for all 50 in this particular superfamily. So in this particular superfamily, your question would be answered with yes. It's not always like that. In fact, it's rather the exception that is so extreme. And we get back to what I said before here. Now I do take all my sequences, whatever the representation of all sequences is, rank them by size of a family, and I asked for how much do my largest family represent the entire sequence space that I'm looking at. And so that's 10,000 here. And what I see is that the first, so somewhere here is the number that I said, the first 300, the largest, 300 largest superfamilies account for, or together have about 50% of all the sequences at that time. These numbers change a little bit. And some I can't remember what the next cutoff is here, but this is the first, I believe the first 20, or I can't remember what the first cutoff here is, but it's the first 20 or 30 families are immensely big. So some of these have 100,000 sequences in them. But you see, this is a very skewed distribution. That brings us back to, in some sense, all 50% of all the sequences fit into 300 folds, into 300 shapes. So this is an extreme way of saying sequence space is big and structure space is small, right? Again, from Janet Thornton and Christina Rango. Cath. Now, Cath still relies on expert information, still has a classification that is intuitive. As a class, it has different levels, and all of these levels make sense, but they're intuitive. Manfred Zippel from University of Salzburg, again, one of those examples, the H index is 27, but if you look at structure prediction, threading potentials are Zippel potentials. Potentials of pairwise energy, knowledge-based potentials, they come from him. So that is something that everybody in the field uses, everybody who predicts structures. So the H index is not always reflecting the impact that you have on a field. COPS, classification of protein structures. And this one here now is completely based on quantified structural comparisons. At some point he called it QSCOP, things change, but essentially this is an automated way to classify structures. And the automated way to classify structures begins with a metric. Remember that I showed you, and the slide is missing here, that in a simple case here where you have protein A, B, and C. So A being similar to B and A being similar to C does not mean that B and C are similar. The metric is violated in the comparison of sequences and structures. And that is a problem that the field has been wrestling with. How could you surmount that problem? What could you do? What the field did for over 20 years was to simply say yes. Mind you, structure comparisons or sequence comparisons are not metric. So that has certain implications. Let's move on and let's work on something. Nobody ever tried to put a metric in there until Manfred came. So how could you do it? Yes, this is the solution. The answer to my question is on that slide. But it is in a way that I'm not entirely sure what your math background is. You may not pick it up in 10 seconds. How could you do it? That's not what I want you to do. Everybody's trying to get the math. And I'm scared that you actually may get it.
Something about length.
Something about length. It has to do with length, ultimately.
Yeah, regarding that A is longer.
Yeah.
Taking this into account.
How can you? Look at the example. How can you? The answer is, yeah?
Just take a subset of A, you vary the length of A and compare it then?
So you move a fragment.
Yeah.
This is somehow is exactly the solution that we all had in our heads, because that is the solution that you, in some sense, you believe in when you say domain, when you say fold, when you say, like lupas, even subfragments that are conserved. It always is somehow a sliding window, and then I hope that, boom, I find the fragment that hits and that has a similarity. But that fragment is still not solving the problem, because that fragment may be here. Well, okay, there may be a second one than these two. So that fragment is defined, the way you move it around, is defined by pairwise, right? No, I'm not countering your argument. But that fragment is not the solution, because it still is a fragment, and the fragment actually, you need a more global view. That would, in this particular case, really would allow you to compare C and B. Let me give you another point. Why does that matter? It matters because in classifications such as CAF and SCOP, you can say that two proteins are related. Or you can say that I do not know whether they are related, or cannot tell that they are related. Cannot tell that they are related does not tell you anything about distance. Two proteins that you cannot tell are related to something, they may be related to each other or not. Being distant from A or B does not tell you anything about C. And that ultimately is the problem with the metric here. In a real metric, what you have is when A is similar to B and A is similar to C, then B and C are similar. That's the triangle inequality. Higher similarity means lower distance. There's a 1-to-1, a 1 minus the other one. And that is missing if I don't have a metric. Now, if I want to classify proteins, knowing similarity is good. But in order to make an image of the world, I know I need distance too. I need to group things that are really not like the other one. Okay, on the level of alpha, beta, other, I can do that. It's a very simple space. In that simple space, I can define not quite a distance, but a binary distance in some sense. But on the level of the fold, this problem prevents me from doing that. So again, what can I do? So the sliding window is not the approach. The sliding window, I believe, is exactly, you have to sort of The solution is extremely trivial. And I believe we all didn't see that because we all thought about the sliding window all the time. I thought about this problem before the publication of Zippo. That is actually not that old. It's relatively recent. It never even occurred to me. It's totally simple. I actually would argue that you guys are better in a position to find the solution than I, because you're less biased by the mistakes of the past. Look, you have to just do the maximal model. In the maximal model, the whole thing starts counting from here. It ends counting wherever the last one is. And now you compile, you normalize by the entire stretch. And you compile the similarity. So since that similarity here is more or less than that, you get a feel for as part of the entire possible alignment. Because now the entire possible region expands everything. And suddenly you create a metric. As simple as that. In terms of comparing structures, we never thought about this because it doesn't make sense. Why would I start counting somewhere where I have no alignment? None of these comparisons fills this space or this space. So it's in some sense wasted space. That's why I believe why we never thought about it. It also evolutionarily appears to not make a sense. That's the simple definition here. That makes the metric, so you take the full length of the maximal length of overlap. Once you have that metric, now you can go out and simply classify by structural alignment the entire database. When you do that, you can then, for instance, go about and find out how much novelty is added in a set of structures that have been added this week to the PDB, to the protein database. Where blue is 0% novelty. Red is 1 to 20%, green is 21 to 50. Real novelty is black. And that is exactly what you don't see. Or when you look at a better resolution, you see black does appear. But not very often. This is over, I can't remember which period of time here. It may be a year that they looked at. But you also see that the vast majority, so this is really a number of domains here, the vast majority of those is absolutely blue. Meaning that with respect, if you classify by structure the universe that you have today, and you wait for this week's release, you get almost nothing new. Essentially nothing new. So every single structural point you already have in your database. Okay? You could not do that without that method. It is an interesting finding. Let's just leave it there. But what it can do is you can parse domains and you can classify domains, and you can then, once you have this all sorted out, get into a method that classifies proteins, has new biological units, because now it's not really a domain anymore. And you can apply the metric, and you can in fact have distances. That same thing also allows you to classify pairs of proteins or interacting proteins. And it's the only tool that can do that. So those are 3 different ways to classify structures, and mostly used by the world is this. This is the most actively researched database. I continue to believe that this is the place to go if you really have a question. SCOP, I believe, has had its days, simply because the database grew so much that CAF took over, and Alexei just got bored looking at structures ultimately. Any questions about the 3D comparison? If not, we begin with alignments.
Taking some architectures like the beta barrels, can I look at proteins and see a barrel and assume it's a kind of similar function? Because like a barrel for things passing by, passing through. So can I take it and assume they could be like have a similar function regarding this simple architecture?
Sometimes you can and sometimes you can't. So again, in the tin barrel, you can look at it and see the active site. They're all enzymes, and you know where the active site sits. It sits on top. You can see that the beta propeller fold. There are flavodoxin. There are a couple of folds where you can look at this the 3D structure and see the binding site. In that sense, this is exactly the yes answer to your question. The no answer to your question is, and that I will only get to in the next lecture. I mean, in the second lecture about protein function, Janet Thornton again, in a paper that she wrote, just let's bring up her face, in a paper that she wrote on the classification of enzymes with Susan Taylor, pulled out examples where you have the same activities, the same enzymatic activity, the same active site, modeled on totally different structural regions. So you look at them, they have nothing in common. What they have in common is they essentially provide backbone for the activity. Okay, they provide the backbone you need in order to get a certain spacing of the cleavage of a bond. And that you can do with two very different structural motifs, and there's just no way. Now that you see that they revealed it, you look at it, you understand, yes, that makes sense. But had you looked at it before, had you only looked at the 3D object, you would most likely not have guessed it. And that is exactly the no to your answer. So can I look at shapes and essentially predict function? And that brings us to the next issue. So structural genomics was a large project that was funded in the US since 2000. And the last round of funding was funded in 5 years cycles, and the last funding cycle is now ending in June. Amount of funding in the ballpark of $200 million per year, and the goal was to essentially simplify structural biology, make structures available, bring structures to a level where structures are cheap, to bring large throughput technology to structural biology. How much does it cost to do a structure by X-ray crystallography? Ballpark? I showed you many figures. How much does one cost of them? Would anybody be shocked if I told them $10,000? Would anybody be shocked if I told them $50,000? We begin to say some shock. What would happen if I said 100,000? Oh, that's too much, huh? What if I said 1 million? So in 2000, most likely the average structure was more than 1 million. One goal of structural biology, or the NIH-funded Protein Structure Initiative, Protein Structure, was structural genomics, was to bring that down to 100,000 per structure. After about 10 years, they got there. The large factories, so there were some large factories or some large consortia financed in the US, each of which got about 10 million per year, and they cranked out structures at an immense speed. So some of those cranked out structures 1,000 a year or something like that, which is incredible. So again, 15 years ago, one is the number of money, another one is typically a structure was a PhD project, 3 years or longer. And suddenly they do 1,000 in a year. There was a revolution, and they did it by robotics. Now, again, one of the scientific drivers was what I said. We want to fill up all these islands, the white islands. So get a structure for everything for which we don't know the structure. What we then very early on got into realizing is, and everybody believed that looking at the structure, we would be able to, from the structure, guess the function. In 2000, I believe very few people would have answered no to your question. Everybody would have immediately jumped up and said yes. All the experts in the field would have, all the structural biologists would have jumped up. And then suddenly we were faced with this situation where we had a bunch of sequences for which we had structures, but we had absolutely no experiment. So we had a 3D structure, but we had no biochemical studies what they actually do in a cell. And these were selected by computational biologists, by us folks, as things that have large families for which we don't have a structure. They were not selected by I know the function does that or that. Rather the opposite. We looked for things that are really wide, where nobody knows anything, but we have huge families. So evolution has repeated that motif again and again, so we argued that must be important. And we don't have a structure, so if we get one, we have a high bang for the buck. We can model many, many, many proteins with it. And then we began to realize very early on that, in fact, it's much more difficult than we thought. That structure alone does not give us as much clues as we had hoped. The next step was to, and that in some sense is what Christine really still does today in Gene 3D, and in fact taking these superfamilies and bringing it down to the level of function again, is combine sequence, variety, large families, and structure, and together you can learn something about function that with only one of the two you could not. And in that way she gets way beyond what PFAM can do and way beyond what in fact SCOP can do. Sorry for the long answer. Anything else? Okay, then we go into the alignment. Since we have only 15 minutes for the alignment story, the one part that I did not have on my slide last time here, the makeup is October 13th. So today will be about alignments, and since we have only 15 minutes, I skipped the slides. Bring them next.